So welcome to the lecture on soil and air quality. I will be your lecturer for this course. My name is Professor Samuel Osu-Jesse. So, for the purpose of this course, permit me to introduce the course properly. So, the course is on water, soil, and air quality. But you realize that the proportion of water on the air surface is so huge that it's the quality of water that ends up affecting the soil quality, because most of the impurities would end up finding themselves going to soil. So we would realize that the water portion would be greater compared to the soil and air quality than water. So water by definition is known to be a colorless, odorless, and tasteless liquid. In terms of the molecular component, it consists of two atoms of hydrogen and one atom of oxygen. And these are joined together by a covalent bond, as you can see on the slide. Now, like I said, water has no taste, neither does it have odor, standard temperature and pressure. And so we say water has a very high specific heat capacity. The specific heat that we talk about here, the amount of energy that is needed to otherwise vary the temperature of substances. But because water has a high specific heat capacity, it can absorb large amount of heat energy before it can get hot. And it also means that water releases heat energy slowly when situations cause it to cool. Now, water's high specific heat ability allow for the moderation of Earth climate, and this assists organisms to regulate their body temperature effectively by way of osmoregulation regulation. Let's look at the different sources of water that we may have. So when you look at the air surface, as you can see on the slide, you have water from the piezometric surface, okay? As by the line indicated in the middle of the slide, we have water from the water table that is abstracted from the belly of the earth. Some is also confined in impermeable area, as you can see on the slide. And there's also some on the top of the confined aquifer. And all these water come together to exhibit what we call the aquifers and wells. And these are the sources of water that we have. These include waters that are found in streams, springs, water that are found in lakes, and even the sea. What is surface water? As the name implies, surface water is water that is found on the surface of the earth that is visible. And this water is any body of water that is found on the earth's surface, including the sea, that is salty found in the ocean, and fresh water that is confined in rivers, streams, and lakes. Now, the body of surface water can exist all year round or for only a short period of time, and this includes rivers, lakes, and streams. The slide you see here are pictures of surface water that are very visible. So one would say that because this water is always on the surface, these are the water that is likely to be polluted. Let's compare them to groundwater. So we say groundwater is a water that is found under the, for the want of a better word, sorry about this, for the want of a better word, found in cracks and spaces in soil, sand, and even rock. Normally, this water, when found, is stored and they can move slowly through geologic formations of soil, sand, and rock, usually called aquifers. But then groundwater is used for drinking by more than 50% of people living in many parts of the world, including the United States. The largest use of groundwater for most countries is to water or irrigate crops to aid in agricultural and other agri practices. So as you can see, the water comes to the surface through aquifers and they move through unsaturated zones and they are normally confined to areas we call streams and other water body holding areas, and these are used for the purpose of agriculture. For the aquifers, aquifers would are normally mentioned in water quality studies, and these are typically made up of gravel, sand, sandstone, or even fractured rock like the limestone. Now, water can move through these materials because they have large connected spaces that make them permeable. Make them permeable. The speed at which groundwater flows is also depend on the size of the spaces in the soil or rock and how well the spaces are connected. So groundwater can be found almost everywhere. And here, the water table may be deep or shallow, and they may rise or fall depending on how many factors come into play. But in heavy rains and melting snow, it's also capable of causing water to rise. And these account for most of the water found in our aquifers. Now, you see, groundwater supplies are normally recharged or otherwise regenerated when it rains or when snow falls and the snow eventually melts and they move into cracks and crevices beneath the Earth's surface. So we say water in aquifers are normally directed to the surface through a natural phenomenon, sometimes through springs, and they can be discharged into lakes and streams. Let's compare them to well. A well is a pipe in the groundwater that is capable of filling um, a space. Now, normally, we've all seen well, some are naturally made, others too are done by human beings. Now, in many areas where water is scarce, people can dig wells that goes to about, let's say, 10 meters beneath the air surface, where water normally moves up. In some instances, too, they use mechanized boreholes to also sink wells. All of this is an attempt to make sure that water is available. But the nearest of materials above the aquifer is permeable. Pollutants, as seen, can easily sink into groundwater supplies, and this can pollute the water. There are instances where you can also find a designed landfill or deep septic tanks, and this can leak. And as they leak, they can also pollute this groundwater, although groundwater is supposed not to be polluted. But if groundwater becomes polluted, then it means it's not safe for drinking. Let's see how much we depend on groundwater for use. Now, groundwater supplies drinking water for more than 50% of the people on the earth, depending on which region you find yourself. But then groundwater also helps grow our food. Groundwater therefore becomes an important component in many industrial processes, especially the food processing industry and even water producing industries, because groundwater is a source for most of this water. And these are able to run through the entire period of the year, from January right to December, even in the areas where, or in times where the weather is dry, there is some level of groundwater that is available for use. And these are so important to be used for our daily use. So we say examples of groundwater are categorized into two in terms of aquifers. Some are confined, others are not. And this is a comparison of surface water and groundwater as you can see on the slide. So in terms of comparison, we say surface water have a composition that is very variable, probably because they are on the surface of the earth, whereas groundwater has a constant composition. Now, in terms of mineral content, we say groundwater has high mineral content compared to that of surface water. But in terms of turbidity, the turbidity of groundwater is low because probably they hide under the belly of the earth, whereas surface water, because it's available, usually turbid. But surface water do have dissolved oxygen and they can easily accommodate fish and other aquatic organisms compared to that of groundwater, which has very low dissolved oxygen and even no dissolved oxygen at all. In terms of hardness, groundwater has high hardness compared to surface water, whose hardness is a bit low. What are the uses of water? Water is so useful as you can see, because I believe even today you have ground water. Some of you have used water for washing your faces and for bathing and for other domestic purposes. Groundwater is also, or water as in general, is also used for agricultural purposes, especially irrigation, livestock, and even those involved in aquaculture. There is also the domestic usage, where water is used for washing our dirty clothes and other activities like cleaning and even bathing. It also has some industrial uses, especially in water and wastewater usage, especially for waste water disposal and even cooling of our engines. Other uses of water include hydropower generation, where water is used to generate electricity, medication, where water is used for travel, recreation, where water is used for swimming and other water sports, and even wildlife and natural conservation in terms of environmental uses. And these are some pictures that I brought to you to help you understand the things that I speak about. Water cycle. One would expect that the water that we use get depleted, but thanks to the water cycle, we always have abundance of water to use. But that doesn't mean water should be polluted. Now, from the slide you see here, normally water from streams during the day would get evaporated and form as light clouds in the atmosphere. In much the same way, water under the water table also gets evapotranspirated through the activities of plants. But when these water are evaporated into the atmosphere, forming smaller clouds, with time, the clouds join together, they become heavy, and they fall as rains, and they go back to the filled as streams and other water bodies, and the cycle continues. And that is how come we normally have water all year round, because they go through this water cycle. Water pollution. Because surface water is always available to use, they are often polluted. And so if they are polluted, we say they are contaminated. A water that is polluted simply means water that is contaminated with some impurities. Substances that cause water to get polluted are known as the water pollutant. And these pollutants are mostly biological and chemical agents. Pollutants can come from our home during our daily activities, sometimes from farms, and even industries. And this polluted water is normally considered unsafe for human consumption, and as much as possible, they must be treated. Now, if water becomes polluted, they can cause diseases like typhoid, cholera, and gastroenteritis, and these have fatal consequences. Water getting polluted can also destroy the ecosystem. They can lead to algal bloom formation in the form of eutrophication, where the water surface becomes very green and aesthetically displeasing. They can also disrupt the food chain. But as a destruction, this can also lead to the destruction of life, especially in both aquatic and terrestrial environment. And these are the consequences of some of the water pollution. Here you see eutrophication, the formation of algal bloom. Now there is some loss of aquatic lives, and there are the emergence of diseases linked to some of these water-related diseases. How do we prevent or control water pollution? Mainly, we need to educate the people involved in these activities. People who pollute our water through mining and other unlawful activities must be educated to know the harmful effects. Education alone may not work. There are instances where we may have to also prosecute people who flout these environmental laws so that we can also prevent water pollution. Now, is water important? Yes. Water is so valuable because 2 million tons per day of human waste are disposed in our water bodies, and these waste would end up polluting our water. Now, we are saying half of the population of the developing world, including Ghana, we are exposed to polluted sources of water, and this can also lead to disease burden. Water pollution alone, research has shown that we are capable of killing about 3.6 million people on a yearly basis, and this is a matter that needs urgent attention. We cannot say water is polluted without talking about plastics and other water pollutants that kill more than 1 million seagulls and even 100,000 marine mammals on a yearly basis. We are also saying that almost 70% of industrial waste are not treated, and these are dumped into the receiving waters and thereby contribute to excessive water pollution. And so we say drinking water becomes a threat. If water is constantly being polluted, then the drinking water is endangered. And we are saying that there must be regulations led by the regulators to make sure that we establish both primary and secondary water qualities, and this must be enforced. So we can have water devoid of microorganisms and other disinfectants, and people can consume them without any health challenge. So that brings us to water quality and water quality studies. By water quality, we are talking about the nature or the quality of water that we bring in our system on daily basis. If you bought water either in a sachet or mineral water bottle, you want to be sure this water is of high quality. So that once you bring them, you will not have any negative health effects. As you may already know, we know water, the earth is covered by more than 300 trillion gallons of water, and that accounts for about 21% of water on the water surface. But it must also be noted that most of this water are not safe for drinking because they are salty. About 97% of water are salt, leaving us with only 3%. But out of these 3%, majority of them are not even accessible as they are locked up in aquifers and in icebergs, in other glacier related areas. leaving us with only 0.4% available for consumption. So even in spite of all the water that we have, less than 1% is available for drinking and accessible, then it means measures must be put in place to make sure that this water is not polluted. And then the one that we drink is of high quality. So as I made mention, these water are held up in glaciers and other polar ice caps, and they are not readily available for drinking. Water quality studies. Those of you working in environmental protection agency and even water protection companies, water quality will come as a very important tool. In fact, water quality is used to assess how polluted or otherwise how clean a given body of water is. And mainly, we look at the physical characteristics of the water, chemical characteristics of the water, and the biological characteristics, otherwise, the radiological characteristics of this water to assess whether they are fit for drinking and other domestic purposes or not. And there is a procedure that we use. But then before you spend time and money to conduct water quality analysis, there is what we call the water quality index, which must be done to even ascertain whether it is worthy of even conducting this laborious water quality analysis. And we say the water quality index is a systematically calculated index or number that represents the overall quality of water for a given community. And this normally informs the individual, the researcher, about the pollution level of water. And they can be used for, or they need to be treated before use. And it takes into consideration both the physical chemical and even the microbial parameters. Now, Demo and ICA conducted a research where they sampled organic city for the physical chemical and microbial analysis of the water samples that they had. In their study, they determined the overall quality of the water quality index using the formula, and they found out that it was 87.81, and this was ranked poor in terms of quality due to the presence of heavy substances exceeding the acceptable limit. And in this case, obviously, they needed to treat or conduct water quality analysis. I must say that water quality analysis is a very expensive procedure because you use labor and you use time, and these are all calculated in terms of cost. So in water quality analysis, you are interested in finding out the pH of the water to ascertain whether they fall within a given acceptable parameter. You also look at the turbidity. Then you look at the total dissolved solid, otherwise known as the TDS, which must conform to values as you can see here. You look at sulfites, look at calcium, look at nitrate, look at arsenic, lead, fluoride, copper, chloride, and the list continues. And these are many. And this individual content that are found in water must be determined chemically, and these can be very expensive. And so before you go into even do this water quality analysis, you must ascertain whether it is even worth it. Now let's start with the chemical parameters. What are the chemical parameters? These are parameters that are a combination of the physical and chemical properties of water that is used to ascertain its quality. In the physicochemical parameters, we are interested in some aesthetic characteristics that include the color, stability, and other properties. We are saying toxicity reactions, such as reactions that compounds and other elements are able to produce can be very detrimental and harmful to living organisms. And we must ensure that before water is consumed, their quality is determined. So in water producing companies and even the beer manufacturing companies, before water is used, these qualities are determined to see whether they conform to the right standard. So we'll be interested in looking at the temperature, pH, electrical conductivity, stability, total dissolved solids, chloride, sodium, ammonium, dissolved oxygen. We will also be interested to know the biochemical oxygen demand, the chemical oxygen demand, the nutrients, and other microbial parameters. Now, there are other elements known as heavy metals that are also found in polluted water, like acidic carbon that also have very negative consequences if they are found in water that is consumed. Now, at this point, you will start looking at the different parameters that are measured in water quality. Let's start with the physical parameters. And the physical parameters, we start with temperature. Temperature, as we know, is the hotness or coldness of a given water body. Now, assuming water is leaving the factory floor where production takes place, this water will normally find themselves in streams. And so here, the difference between the temperature of the stream and the temperature of the water leaving the factory floor must not change. Assuming the temperature of the stream is, let's say, 20 degrees Celsius, there are some aquatic organisms that are used to this temperature of 20 degrees Celsius. And so if we are discharging water of temperatures going beyond 30, 35, and even 40 degrees Celsius, it means we are going to offset the temperature of the stream. And here, aquatic organisms that are used to these temperatures can be killed. Now, changes in temperature can also affect reaction rates and other suitability of water for beneficial use. So it's important that this water is measured. If they are measured and their temperature goes beyond that of the receiving water, then it means it's going to cause problems. The instrument we use to measure these temperature are the thermometer, but these days there are other probes that are specially designed that can be used for that. Now, both the increase or decrease in temperature could also have dire consequences, just as I just said. And they can also affect the rate of biological activity, especially the growth of microorganisms. And this can be affected negatively or positively, depending on what type of microorganisms that are in there. If there are beneficial microorganisms that are needed by these aquatic organisms, they are destroyed. If there are also other organisms that could also cause other activities, they're also affected with these changes in the temperature. pH. Normally, you would realize that pH refers to the power of hydrogen or hydrogen ion concentration in the given water body. So normally on the pH scale, we have pH of 1 to 7 being acidic, 7 being neutral, and then 8 to 14 being basic. Now, different water bodies have their pH regulations. And so if you are discharging an effluent with a pH range that is different from the receiving water, it's going to change the pH of the water that is receiving the discharge. And this can also affect the quality in terms of the pH. Once the pH is changed or varied, other and taste properties of the water can also be influenced, and this can also control equilibrium concentrations, neutral and ionized forms of water. Now, there are different probes, as you can see, that are used to measure the temperature and pH. And these days, there is even multi-purpose probes. And in these multi-purpose probes, they can measure pH, salinity, temperature, conductivity at the same time. How is it measured? You put this endpoint of this probe with water collected in a container. Once you power the probe and you set it for temperature detection and you put it in, it gives you the temperature and the other readings that you so much need. And this can give you a rough idea about the quality of the things that you have. These are more examples of probes that are used to detect the pH and the temperature. Stability. When we talk about stability, we are talking about the amount of suspended matter in water that can also change the color. If you have a given water body and it's colored, then it means the light transmission ability of the water is also affected. And this gives an indication of water that is polluted. Now, the unit for determining the turbidity is the nephometric turbidity unit with the nephometric turbidity probe. Now, this measures the clarity, or otherwise the cloudiness of water samples given. The color of the water changes because of colloidal particles that may be present in water. These days, as you move through towns and cities, you realize that the color of most surface water, particularly streams, are changing from colorless to coffee brown. And this is as a result of the position of colloidal particles from illegal mining activities. Ordinarily, water that is fit for drinking must not exceed 5 nephometric turbidity unit, that is NTU5. But these days, because of climate activities, some water bodies are polluted with turbidity, nephometric turbidity units of more than 14,000, 15,000, and this makes it very difficult for treatment. And as they also occur, they also affect aquatic life, and the water is not fit for drinking. The instruments that are used to measure the stability are many, and they include the Celsius disk that is normally dropped in the water body, and they have readings that corresponds to the turbidity units. Sometimes too, we also have other probes that once you insert into water bodies, it can give you a rough idea the turbidity of the water that is given. Water with very low turbidity unit means that it's fit for drinking. But water with turbidity units outside the values recommended by the regulatory bodies such as the EPA means these water bodies is not fit for drinking. And these are some of the probes that can be used to determine the quality of water by way of turbidity. Dissolved substances. Water can also be polluted based on substances that are dissolved in them. Sometimes you can have a very clean looking water with high dissolved solid. This is because dissolved solid cannot be seen with naked eyes, and they are termed TDS. This is made up of organic and organic matter in water, and these mostly come from runoff discharge, and they impact negatively on the quality of the water. They are normally made up of carbonates, bicarbonates, chloride, phosphates, nitrates, and sometimes they can even have nitrates of calcium, sodium, manganese, potassium, organic matter in terms of salt and other particles. And depending on the amount that is present, they can affect how palatable given water body is. Normally, they shouldn't exceed 500, and a water with TDS of less than 500 is considered perfect for drinking. And those that goes very high above 1,000 milligrams per liter is also considered unsafe. Now, there's a correlation between the TDS and turbidity and hardness, and these are all linked to quality of drinking water. Therefore, we expect that high amount of TDS, once present, could be because of the presence of other carbonates present in drinking water. And the TDS is also measured using the TDS probe. This is a sample of a TDS probe inserted in the water collected in a glass, and be able to give the readings on the meter as shown. So this is another schematic detection of TDS using another probe, but the probes come in many forms, and the M is attached. There are other experimental means of determining the TDS if you don't have a probe, and there are some also calculations. Now, to determine experimentally the TDS without using a probe, you need your vacuum pump, you need your Koch crucible, you need your graduated cylinder, you need your desiccator, you need your analytical balance, a filter paper, and a water bath. Normally, you can determine the TDS experimentally by making sure that your Koch crucible is well washed and dried in a hot oven for at least 1 hour, after which they are cooled in a desiccator. Then you weigh the empty crucible using an analytical imbalance. Analytical balance because this is a balance that can detect slight changes in weight. Then you measure about 200 mils of water sample, and then you filter them after shaking them vigorously. Then you take 100 mils of the filtrate and measure them using your crucible. Then you evaporate them to dryness. Then the samples that you have dried in an oven is kept for about 1 hour to make sure all the water content is gone. Then the drying is repeated until you don't obtain any change in weight after 2 consecutive or 3 consecutive measurements. Now, all you're interested in is that you suspect that the filter paper would be used to filter all the total dissolved solids onto the filter paper. Here, the idea is that you measure the filter paper before use, before you do the filtration. And then after the filtration, you dry the filter paper and make sure all the other components are also gone. And then you measure them again. And then when you measure them again, the difference between the weight becomes the weight of the dissolved solid, which you calculate by dividing the mass of the residue by the volume of sample you have. And that gives you the total dissolved solid value that you are looking for. But these days, these calculations are not done. They are determined using the probe, which is much, much easier. Once you set the probe, you're able to get your reading instantly and you can do your permutation. Color. Color is also another parameter that determines whether water is polluted or not. And it's a result of different wavelengths that is not absorbed by the water itself or particulate or dissolved substances that are present. And these are measured by the Logie-Baud visualizer disk. Now, you expect that water that is fit for drinking has no color. But these days, when water gets polluted, they are able to impart color, and they are supposed to be measured using the appropriate instrument to make sure water is of good quality. So there's solids, minor solid. The minor solids can also be found in water, and these are a collection of organic and inorganic materials of different sizes and particles that could be present in a given water body. Here, we normally conduct routine test, and this routine test give an indication of the performance of conventional treatment processes and the need for water, especially effluent, to be filtered if they are supposed to be reused. And these also give an indication of the quality of water that we have. We see suspended solids are not supposed to exceed a certain figure, and we also use different probes to determine these suspended solids. Now, we can also have total suspended solids, and these are abbreviated as TSS, and they refer to waterborne particles that exceed 2 microns in size. Now, here we are saying any particle that is smaller than 2 microns is considered total dissolved solid. Here, majority of the total suspended solid comprise of inorganic materials. However, algae and bacteria may also be considered total suspended solid. Now, while some of them settle at the bottom of water sources, there are other total suspended solid that will still float on water surfaces. They will remain suspended, and their presence will affect the clarity, and they can impact on the quality of water as may be determined by water quality studies. And here also, they also have probes. These days, these probes are so much available, but they can also be determined experimentally, as you can see. So much the same way, we take a given volume of water using a filter paper. We filter all the suspended matter, then we subtract the weight of the filter paper after filtration and drying from the weight of the filter paper before filtration, divide them by the volume of water used, and that can give us the total suspended solid. But you can also determine them using probes that are also direct. And this is experimental determination of what I just explained. Salinity. Salinity can also be a factor, it's also a factor that determines the quality of water. So in salinity, we say the total amount of organic materials dissolved in 1 kilogram of water when all carbonate has been converted to oxide. That is not all. When all bromide and iodine has also been replaced by chlorine, and when all organic matter has been completely oxidized. And here, the presence of salt can also affect growth in the following ways, especially osmotic effect and also specific ion toxicity caused by a concentration of individual ions. Here we can conveniently say that salinity is also the amount of dissolved salt present in a given water body. Now, water that is high in terms of salinity, we say magnesium, sulfate, calcium, and other ions could also be present in small concentrations. And this could all impact on the salinity. Salinity is physically measured with a salinometer, and this is capable of calculating the amount of weight of salt present in a specific volume of water. But they can also be measured with the salinity probe, where once you insert the probe, the salinity can also be recorded on the probe. Now, we can have salinity measurements recorded in both ocean and freshwater, and all of this gives an indication of the total amount of some dissolved chemical present in the given water body. EC, or electrical conductivity. As the name implies, electrical conductivity is the ability of water to transmit electrical current, and this serves as a tool to determine water quality. Normally, all of these indicators depend on the country of origin. There are some values that are given. Now, the value is such that the water quality must fall within a certain domain. In Ghana, EP has a certain range within which the electrical conductivity of water must not fall either below or above. They must fall within the range. If the electrical conductivity is very high, it means it can easily conduct electricity, and such water is not good for drinking. Likewise, if it's also below a certain threshold, it will also not be appropriate for drinking. And electrical conductivity is also measured with an instrument known as the electrical conductivity meter. Once you insert it into the given water body, it can give you the reading. Let's talk about dissolved oxygen. As the name implies, dissolved oxygen is oxygen dissolved in water. Normally, when water from industries is made to move into receiving waters, they must not have dissolved oxygen values that are too low. This is because normally streams and other surface bodies have dissolved water, and these dissolved water are water that are being used by aquatic organisms. If a given water body has very low dissolved oxygen component and they are being discharged into a given water body with high dissolved oxygen content because they are able to support aquatic life, then it means they will affect the life of the aquatic organism. In other jurisdictions too, some water also should not have dissolved oxygen, especially water that is used for other purposes. And so as a matter of water quality indicator, they must be determined. Now, if you have a stream and you realize that aquatic water bodies are also dying at regular intervals, then you, the investigator, must also use your probe to measure the dissolved oxygen content to see whether it is high enough to support the aquatic life. And here also, you can calculate it using either the Runkel method or you use the probe. But these days, there are a lot of probes around that helps one to determine dissolved oxygen content. That gives an indication of the oxygen content in a given water body. And this is a probe, conductivity probe, that can also be recalibrated to measure the dissolved oxygen. And this is essential for all aquatic life. Dissolved oxygen content can also affect the temperature, pressure, salinity, and other photosynthetic activities of organisms like algae and other organisms that are present in the given water body. Biological oxygen demand, otherwise known as the BOD. The BOD is a measure, or normally the BOD5, we say the BOD5, the 5 is subscript, but not as shown here. So it's the capital BOD with small or subscript 5. We say it's the measure of the quantity of dissolved oxygen used by microorganisms in oxidation of organic matter. If you have wastewater and you believe microorganisms present in this wastewater can otherwise dissolve or oxidize organic content of the water, then the BOD must not be too high. Normally, microorganisms utilize dissolved oxygen, especially aerobic microorganisms, utilize oxygen that is dissolved in water so they can oxidize polluting biodegradable organic matter, thereby giving an indication of the pollution load present. But if the BOD is very high, it means the water is very much polluted. And microorganisms to have challenges trying to treat it by oxidizing pollutant. And this can also be measured experimentally by determining the POD for the first day, and then you keep your samples in a dark room, and you also calculate the POD for the fifth day, all maintain at an incubation temperature of 20 degrees Celsius. You can find the difference. And this gives you an indication of how polluted your water is. Now, we say the POD, or the biological oxygen demand, or the biochemical oxygen demand, gives an indication of contamination in the water, especially by waste discharge and decomposition of organic matter. We are saying an increasing amount of POD indicates that higher amount of organic matter is present, and so it's not easy to treat such water bodies. And these days, we also have the POD probe that can be used to measure the POD for the first day. Then you keep your samples in an incubator maintained at 20 degrees Celsius. Use the same probe to determine the POD after 5 days. Then you find the difference. That gives you an indication of the biological demand. That gives you the level of pollution of your water body. Now, the equivalent or the chemical equivalent of the BOD is COD. Normally, BOD can be computed after 5 days. Why? This is because you need to compute the BOD of the first day, then keep your sample and incubator for 5 days, maintain at a temperature of 20 degrees, and then you compute the BOD after the 5 days and you find a difference. But if you have a given water sample and you want to determine the extent of pollution, you can use the COD, which is the chemical equivalent of the oxygen content of the water bodies, and this can be obtained within 2 hours. So we say the COD is also the equivalent of organic material in your water that can be oxidized chemically using dichromate in acid solution. It is more convenient as the results can be obtained within what we call 2 hours. Now, in addition to these chemical parameters, we can also determine chemically nutrients such as phosphates, nitrates, and ammonia. This can all be found present in a given water body that can be determined to determine the extent of pollution. There is also the heavy metal part that can be in the form of arsenic cadmium that must be detected. Now, there is also chloride found in water. Chloride in water might also result from the release of organic waste by microorganisms dwelling in it. And this can also pollute the water, which level must be determined chemically to determine the extent of pollution of the water bodies. Here, you can also use what we call experimental determination, where you use silver nitrate solution, potassium chlorate as indicator with the help of the still water. And this is the chemical processes. Here, I must also say that for environmental scientists who would use these processes, this is where your chemistry would come in. And here, you use some chemical processes to determine the level of chloride in the water. That gives an indication of how polluted the water is. There is also similar processes where you determine ammonia, and ammonia present in the water give a strong correlation with the presence of microorganisms due to the release of organic or metabolic waste substances present. And a water body with high ammonia content means that the water has high level of microbial contaminant present as they continue to release metabolites, and this can impact on the ammonia and even the odor. And this is how ammonia is also determined experimental. And here, I want you to rely on a knowledge of the environmental chemistry you did to help you determine this ammonia. Microbial assessment. Microbial assessment is the last portion of the water quality analysis indicator that we will look at in this lecture. Here, for those of you who did biology, your knowledge of pathogens would come into play. Now, before we go into this, let us take some time and look at some definitions. So the microbial parameters of water, we're looking at pathogens, and pathogens are disease-causing microorganisms. So here, we are talking about disease-causing microorganisms that are found in water. There may be terms like infected dose that you also come across, and this is the amount of pathogens that is required to establish an infection in a given water body. So the amount of pathogens is normally considered as colony-forming unit or particles in terms of viruses. Now we are saying if a person were to be exposed to exactly one copy of SARS-CoV-2 virus or a single Bartonella species, is unlikely to cause an infection simply because the body's immune system can easily take care of them. But these pathogens, once they come in larger numbers, then they can cause the disease. And so we say the maximum amount of particulate matter or microorganism that is able to elicit a clinical symptom to an individual is known as pathogens. And so we need to determine this level of organisms in a given water body to determine whether it is safe for drinking or not. Now here we say organisms with minimum infective dose, which means the minimum level below which they cannot cause diseases. For Salmonella, we have 10,000 to 10 raised to the power 7. For E. coli, that is a pathogenic E. coli, we are looking at 10 colony-forming units to even 100. For Shigella, we are looking at 1,000,000 to even 10 raised to the power 8. For Giardia lamblia, we are looking at 10 to 100 seeds. Ketosporosis, we are looking at 10 seeds. And Amoeba histolytica, we are looking at also 10 seeds. For Hemorrhage, we are looking at 10 eggs. For viruses, 10 is enough to even cause an infection. How do we determine biological characteristics of water? In biological characteristics of water, we're looking at two main attributes. We're looking at the specific microorganism present and the biochemical substances or disturbances that may occur. And this can give an indication of pollution of water. Let's look at specific microorganisms. Now, water that is polluted is suspected to contain fecal matter, fecal streptococci, enterococci, and E. coli. In addition, their dissolved oxygen is supposed to be very low, BOD is supposed to be very high, COD is supposed to be very high. Same for nutrients. Now, if you are looking for biological properties of any given water body, these parameters are the parameters that usually will come into play. Total coliforms. Now, because it is practically impossible to determine the presence of all of these at the same time, we use a much convenient method known as a total coliform. Now, total coliform is conducted on a given water body. If the total coliform is positive, then one has to go further to do other tests to find out the actual microorganism that is present. Now, we are saying they are the most common risk of human health associated with wastewater. And this gives a rough idea. This is because according to Metcalf and Eddy, a research conducted some 20 years ago, they are saying human beings discharges close to 4 billion coliform bacteria per day, in addition to other kinds of bacteria. And so looking at the amount of living human beings that we have, and discharge that we normally do, then it means water can be polluted with this and they must be detected. Now, the main reason why we normally use the total coliform in determining the quality of water is that we cannot practically, you know, look at the individual organisms. And this is because there is a discharge of domestic waste directly into drains. People are openly defecating. There are municipal discharge without treatment. Hospitals are discharging their waste without treatment. And even when gray matter are treated, they are poorly treated. In areas where we build treatment system, leachate from these sanitary fields end up polluting these water sources. And so there is the need to, otherwise, you just hold on, let me get my charger. Look like my laptop is running down. So there is the need to, otherwise, determine the microbial quality of this water to ensure that its quality is good. And once it is used, diseases cannot manifest. So normally we use E. coli because detection of all possible pathogens in water is rather complex, expensive, and a little and time consuming. And so normally in total coliform detection, we are targeting the presence of E. coli. If E. coli is present in a given water body, then we can conveniently say in terms of microbial, it's contaminated, and so we can go further to look at the other biological properties. Then the reason why we use E. coli is that, just as I said, direct testing in practicals. Other pathogens that are found are normally found in low numbers, and so detection becomes difficult. And most of these can't even survive under living host. And so once they are out, they can also grow. And so we use E. coli as an indicator of choice. And these are the reasons why they are employed. Now, E. coli is used because it is useful for all water types. E. coli are present when other pathogens are present. They're also absent if other pathogens are absent, and it gives a correlation or degree of pollution, easily detectable than pathogens. They can survive longer than pathogens, and they are not dangerous to also work with. So the other bacteria that are used as indicators include total coliform, fecal coliforms, and E. coli. If E. coli, like I said, is present, then we are looking at the fecal streptococci, enterococci. Then we can even go a step further to look for spore formers. These are, but I must also be quick to say that there are problems with the coliform detection if not done very well. Sometimes there can be false negatives and there can also be false negatives. So it can be false positives and false negatives. False positives are when you are saying organisms are present, but in real terms they are absent. Or sometimes they can be present and you are saying they are also negative, and so they must be done very well. So like I said, you normally will conduct a total coliform for a given water body. If it is positive, then it means definitely E. coli and fecal coliforms are present. So you can do your test and then total coliform. Normally, when you do microbial tests, which you go through the processes, you are supposed to see a gram negative bacillus rod-shaped total coliform, and this normally will ferment lactose at 35 degrees Celsius. This is because these organisms are found in both cold and warm-blooded animals, and once they are present, it gives an indication that Klebsiella Enterobacter, Serratia, Citrobacter, and even Edezella are all present and they must be investigated. There's a lot more information to add to what I just said. Let's look at the method of identifying bacteria. Method of identifying bacteria. We can use the multiple fermentation tube method, the membrane filtration method, enzyme filtration method, gram staining, specific media method, or even PCR. Let's look at the bacterial filtration method. I will try as much as possible and do some practical sessions with you. I will arrange with my technicians and you would have one Saturday to do the practical sessions. Now, if you have water and you want to determine the number or the presence of microorganisms, you can use what we call the membrane filtration method, where you would have a given volume of water in a glass slide, then you find the 0.45 millichrome filter paper. You filter the filtrate to the filter paper. Once you are done, then you put the filter paper on on a media. Once they grow, then you can also count the number of growth. That can give you a rough idea. Or there's also the most probable number method, where you can estimate bacterial numbers based on color change or amount of gas produced through specific bacterial metabolic process. You will go through all of that. Let's start with the membrane filtration method. Assuming you have a known volume of water, let's say 200 mils, and you want to determine the microbial load, you first of all will have to use this instrument we call the filtration unit. Normally, a pump is connected to this side of the filtration unit. You put this canister on top of this bench here. Then you lay your filter paper on top of this canister. Then you cover it with any of these. These are different canisters that are used. This is not to say that all the bench have all of this. No. You can either use this, but it will give you a rough idea how much volume of sample you have. They sit on this canister with a filter paper put in here. The pump is put on, the water is filtered, leaving the organism on the surface of the filter paper. But because the organism on the surface of the filter paper, you cannot see them, you may have to culture them. And once you culture them, they grow. And using the appropriate microbiological method, they are identified and then they are counted, and you have a rough idea what they are. In the lab, this is how they look like. So this is your pump. This is your basin. The pump is connected to the base of the conical flask. The filter paper sits on top here. The sample sits in here, which you pour. It is filtered. Then you culture, and you have your results. Now, how do you calculate your results? You count the specific colonies. If per your work, you are looking for E. coli in terms of total coliform, that gives you either blue or red colonies. They are counted, and once they are counted, they give an indication. There are instances where your sample may be so much polluted that the colonies that you find are more than 200 colony form units, in which case you are not able to count them very well, even using the colony counter and magnifying glasses. Then you use what you call the serial dilution. You serial dilute the samples to ensure that the water is not as polluted as you expect. For instance, you did your initial filtration and you cultured, and this is what you saw. Clearly, you will not be able to count the bacteria that is here. So there is a method that we call shear dilution. How? You will prepare 9 mils of distilled water in these portions. So you prepare 9 mils of distilled water, 1, 2, 3, 4, 5. You put them in racks. Now you shake vigorously your mother sample. You take 1 mil of the water sample, you add it to the 9 mils of the water sample to make it 10 mils. You shake it. Then you take 1 mils of that. You take 1 mils of this into 9 mils of the second 9 mils. You shake it. That becomes, so when you take 1 mil, 9 mil of this water, this solution you have is 1 in 10. It is still polluted because you still cannot count. Then you take 1 mil of this 10 mil, add it to the 9 mil of this still water. This means that this solution is 1 raised to the power 2 of this. You keep diluted until probably you get to 10 raised to the power negative 5. Now, with 10 raised to the power negative 5, what it means that your water has been so much diluted so that now you're able to count. Now you find 4 different colonies on this plate. Then you do the back calculation. Now, if 4 colonies was in 10 raised to the power negative 5, therefore, what will you get if you have 1? Then you can do the back calculation and you'll be able to estimate the right proportion of colonies that you have. You also would have the opportunity to do the practical sessions for this as well. Now, the problem we have here is that sometimes you can have a lot of these colonies, but using the back calculations or shear dilution, you'll be able to have a rough idea. Sometimes you can also determine the fecal coliform using other media. And here you collect the samples, the sterile media, you prepare shear dilution as required, you do your filtration, then you also culture them on a media, usually using the chromocoat in a small petri dish, maintain at 70 Celsius for between 18 to 24 degrees hours. And then once you see pink colonies, you can say they are E. coli. Once you see blue colonies too, you can also say they are other E. coli. But once you see bluish green colonies, then you can commonly say they are Salmonella. A creamy colony would indicate Enterobacter. When there are more than 80 other colonies on a small petri dish, we are saying you can discard them and then, you can probably do shear dilution or you can even use allopathic antibiotic agent so that you can also change the narrative and then you can have a rough idea what you're looking for. All of this is to make a lot of meaning to you once you are able to do the laboratory sections, which I'll plan for you. This is how shear, let me see there. Okay. So Gram staining technique. Gram staining technique too, you also will do them in the lab where you'll be able to see whether a given microorganism is a gram positive or gram negative and what have you. Bacteria found in surface water. For surface water, these are some bacteria are likely to find, diseases that they cause, symptoms that they also exhibit. These are more pathogens found on fresh produce, protozoa found in surface water, Giardia and Cryptosaurus is found in surface water. Now, per the drinking water standard, when you go to EPA and other regulatory bodies, these are the maximum allowable limit by which these chemicals and parameters must not exceed. Now, assuming you conducted your study and you realize that the nitrate levels are more than 10 milligrams per liter, then you can say it is polluted. Same for chromium, fluoride, cyanide,
mercury, and ammonium. Now, in terms of pH, water that is accepted for drinking must lay between 6.5 and 8.5. Any value outside this means the water is not wholesome for drinking or not potable. Same for color, same for turbidity, total dissolved solids, manganese, and the list continues. For the microbial parameters, water ought not containing any of these. So this is where our study on water quality will take us. I must admit that this course will need a practical sessions, which I'll arrange with my TAs for them to have. I'll also arrange so that probably we will meet either face to face, so that if there are questions that needs to be answered after watching the video, then we can discuss. Thank you.